My name is Robin. I'm a postdoc with Rob. In the Young Lab, I'm working on cholesterol transport in our cells. So the reason for this is mainly because of this disease, named Niemann-Pick type C disease, or NPC in short. So today in this short presentation, I will tell you something about NPC disorder. I will also mention some important proteins we're working on, which are involved in endosomal cholesterol transport, or the process of cholesterol traveling through the endosomal membranes, which has strong implications for NPC disease. So firstly, I'll just start from introducing cholesterol. As mentioned in this morning Annika's talk, we all know that cholesterol is really good in our body because cholesterol is required for membranes, so we cannot survive, we cannot live without cholesterol. And cholesterol is also be used by our cells to produce some important molecules. Some important molecules such as steroid hormone and bioaccess. So on the one hand, cholesterol is really important, it's absolutely required by our body. So on the other hand, when cholesterol metabolism goes wrong, there will be some big problems. For example, cholesterol has been linked to so many diseases such as cardiovascular disease, high blood pressure, peripheral vascular disease, Alzheimer's disease, stroke, diabetes, and even cancer. In this talk, I will tell you some another disease, which is Niemann-Pick type C disease, which is also closely linked to cholesterol. NPC disorder is a genetic disease. It usually affects kids. It's not common, it's rare, but it's really devastating. The kids suffers NPC disease, they normally die in their teens. Currently, there's no cure for this disease. So what happens to those patients? So the most serious problem for these patients is that cholesterol cannot be properly transported in their cells. So when we use Philippine fluorescent dye, which specifically binds to cholesterol to stain the patient cells, you can see that massive accumulation of cholesterol in the patient cells, which is totally different from the cells from a normal person. So there are two proteins. We all know that there are two proteins which has been shown to be responsible for NPC disease. One is the NPC1, another protein is called NPC2. So you can see from here, the NPC1 is a fairly large protein. It's localized to the membrane of the later endosome lysosome, or LE/LY. So while NPC1 localized to the membrane, NPC2, another much smaller protein, soluble protein, localized into the inside, the LE/LY. In LE/LY, NPC1 and NPC2, they work together, transporting a large portion of cholesterol to the cell organelle called ER. So in the ER, there are many other proteins, so they can sense transported cholesterol to the ER. So if the cells has low levels of cholesterol, this protein can work together to get more cholesterol for the cells. If there's too much cholesterol, this protein again can function to lower cholesterol levels. So it is pretty much important for the cholesterol transport from LE/LY to the ER. If for some reason, either NPC1 or NPC2, any one of them is deficient, cholesterol cannot get out of LE/LY. Instead, they accumulate in this compartment. So here is another example of NPC phenotype. So this is the Philippine staining of the normal Chinese hamster ovary cells. So if cells lose functional NPC1, you can see the massive accumulation of cholesterol accumulate in these dotted structures, which is LE/LY, shown here. Here's what's happening inside LE/LY. This LDL particle carried cholesterol by the cells obtained from the blood. The cholesterol released from LDL is bound to NPC2, and NPC2 transport cholesterol to the NPC1, and NPC1 then insert this cholesterol to the membrane of LE/LY. And from there, cholesterol is transported to the ER membrane. Currently, what exactly happens here is still unknown. So one of the goals of our project is try to understand how MPC1 function to move cholesterol from LELY to the ER membrane. A couple of years ago, we identified this protein called OP5 as a MPC1 interacting proteins. We proposed that OP5 interact with MPC1. By interacting with MPC1, OP5 may directly extract cholesterol from the LELY membrane and transport the cholesterol to the ER membrane. So in support of this model, we found that if cells doesn't have enough OP5, you also see the cholesterol accumulation phenotype, just like the cells does not have enough MPC1. Later, we paid attention to another protein called HRS, because this protein has been shown to interact with OP5. If cells doesn't have this HRS protein is downregulated in the cells, cholesterol for somehow cannot get out of LELY either, even though the MPC1 and MPC2 appear to be normal. So here's the immunofluorescence image showing that what happens in HRS knockdown cells. So in blue color, you can see the accumulated cholesterol, and the green color, which is a membrane marker, LAMP1. So red color shows MPC1. So you can see that the blue color cholesterol is encircled by both LAMP1 and MPC1, so suggesting the cholesterol accumulates inside LELY. Again, in the HRS knockdown cells, here we show the green color, which is MPC2. So MPC2 nicely overlap with accumulated cholesterol, which is also encircled by this MPC1 protein, indicating that MPC2 cholesterol is localized inside LELY. We then were interested in another protein called VPS4 because many years ago our lab shows that in the yeast VPS4 interact with O7, which is yeast of 5. VPS4 has also been shown to interact with MPC1, and recently we found that if VPS4 was completely depleted in the cells, you also see this defective cholesterol trafficking, as indicated by this Philippine staining. Again, as shown in the previous slide, we show that in VPS4 knockdown cells, MPC1, MPC2 localization, the expression appear to be normal. So based on our findings, we propose that MPC1, OP5, HRS, and maybe other proteins that form a protein complex, which function to move cholesterol from LELY to the ER membrane. And this VPS4 just function to break this complex and to recycle these components. So in summary, our goal is to figure out the exact molecular function of MPC1 with respect to the cholesterol transport. We are aiming for find clues to the treatment of MPC disease. And more importantly, because MPC1 has been linked to, say, obesity, type 2 diabetes, and viral infection, so we believe that our studies will have strong public health implications. So here is our ongoing projects. We're trying to further characterize the interaction between MPC1 and these other proteins, and we are trying to link cell signaling and MPC1 expression. I would like to thank Rob for supervision and the young lab members, especially Abdullah, who did some work on HRS and VPS4, Emma, who is still working on MPC1, Andrew for continuous support and guidance, and the DOS lab and cross lab for sharing reagent and equipment. Thank you all for attention.